Macrophages are immune cells that have a role in killing different microorganisms such as bacteria and viruses. They also remove foreign particles from the body, and they have other roles in removing dead and diseased cells. And through the molecules that they secrete, they can also foster tissue repair, which is, for instance, very important during wound healing. So in a healthy body, they also have many different functions. Because the tumor tissue is not composed only of cancer cells, and there are many immune cells there as well. And macrophages are quite abundant in solid tumors. And what is, we can say, also fascinating, but maybe also scary, is that they can have very different roles there. They can promote killing of cancer cells, even kill cancer cells themselves. And if they are exposed to different stimuli, they can achieve a different functional state where they actually help tumor to grow and to metastasize. So they play an important role both during cancer progression, but also during therapy. So they can help the therapy to go better, for instance, in a certain type of macrophages during immunotherapy, or they can make the response worse in the patient, all depending on which functional states they are in. So what we are doing is that we generate macrophages in the lab. We expose them to stimuli that is important and present in the tumor. And we try to achieve different functional states. So we expose them to the stimuli for which we know in advance that they do something to macrophages. And we also expose them to the complex that we have from patient tumor tissue. And then what we are doing is that we connect them with the macrophages from the patients based on the molecular profiles. And we generate different molecular profiles of these macrophages in the lab. And we investigate them in detail. So we put a lot of emphasis on data analysis. So we are not just cataloging everything that we measured, but we really try to see how these measured proteins and genes are connected and how the signal is flowing from this stimuli that we give them, where we try to mimic all the complexity in the tumor, which is very difficult, but at least to have something that's useful. So how this signal is flowing from the stimuli that is received through the functional changes and through macrophages exerting a new function. First, with these molecular profiles that we are generating in the lab, so we are linking them to those that are found in the tumors through molecular profilings of macrophages in the tumors. So we try to find the links. Then one can, of course, say which ones make, based on statistical associations, predict some roles in the clinics. But for us, concretely, what we can contribute are these signaling maps in the cells that we could study in detail. And then this might possibly give open opportunities for finding new therapies, which we call rational therapy design, because we know actually what is happening there and how it functions. This is, of course, wishful thinking, but what it can also do is just to provide the context for the therapies that others might have idea for. Because we can say, okay, depending on the macrophage functional state, the therapy might act differently. So we are also giving context to the others. We will share all the data with the community. But in the best case, even just these maps that we have might suggest something specific that might target specific macrophage populations. And this is highly valuable because there is an intense research going on, but at the moment we still don't have clinically approved drugs that target macrophages in cancer. If I want to give a big picture, the medicine is in general moving towards also having stronger collaborations with data scientists, data analysts, because this is opening new opportunities. In our concrete case, this happened actually through a common friend who is a computational biologist, the same as me, and he worked the same places in Cambridge and ETH as me, but then moved to clinical bioinformatics, where he worked together with Bettina. And he was aware that both of us have a strong interest in very similar research questions, but we are approaching them from different angles. And he saw there is an opportunity for a high complementarity there, and he brought us together. He put us in touch with another, and then we met. And I really liked a lot also her approach, because what she always emphasizes is that we need to have solutions that are working in the clinics, not solutions that are very smart but are only working in the lab. And she has this very practical view, and I can also come with this of really understanding things on the molecular level, what is happening there. So I think there is a good complementarity. For us, it's a great help for this project, because it really started from something that looked a bit like a dream for all of us, for both of us, what we would like to do, and we think it could have a huge implications. So this is a very, very nice starting point, because so far already we could show that we could link some of these states with those in the patients, and that with the new technologies such as phosphoproteomics, we could also come a step further to finding new regulators. So it has a potential, and I think with this money now, when we do a bit broader experiments, we will be in a great position to actually get even a further funding and to move this a step further, the whole project.